Welcome. My name is James Wormold. I'm a writer here at Europa Science, publisher of Electro Optics. In today's web panel, we'll be discussing the latest developments in laser applications for biotechnology and medicine. We'll be exploring the innovative ways in which laser sources are optimizing these sectors and the resulting commercial applications. Joining us in the waiting room to discuss the topic, we've invited two of the industry's leaders to share their insights and experiences with us. So please welcome Giulio Cerullo, Professor of Physics at Politecnico di Milano, and Brian King, Principal Optical Systems Engineer at Starfish Medical. Welcome, gentlemen.
Good morning, or afternoon, I suppose.
Good afternoon.
Yeah.
At this stage, I should point out that this web panel discussion will be available on demand alongside others in the Electro Optics Photonics 100 series on the website for 12 months after today, in case you'd like a recap of any of the information. But moving back to the present day, I'll be throwing up some questions for our speakers to grab. And if you're watching live, by the way, there's also an opportunity to type in your own questions in the Q&A box for me to put to Giulio and Brian. So I can see our speakers are ready and raring to go. So I'll send it over to them. So Giulio, the first question, let's get down to the nitty-gritty straight away with you here. Tell me about some of the biggest challenges you've faced while overseeing laser-based applications in biotech and medicine.
Yeah, so let me start by saying that lasers are key enabling technology, both in, say, biotech, but also in laser diagnostics and therapy. And they have been so ever since the invention of the laser in 1960. Immediately, people realized that differently from natural light sources, laser emit very peculiar radiation. So they are monochromatic, so they emit radiation of specific wavelengths, and also they have a spatial coherence, so they can be focused down to very small spots, and so they can reach very high light intensities. Now, immediately it was realized that lasers can also be pulsed, so they can generate very short flashes of light of a duration initially picosecond, nowadays even femtosecond duration. So this allows really to concentrate large amounts of electromagnetic energy into very small volumes, resulting in this extremely high peak power and a really peculiar regime of light matter interaction. So the first experiments were, say, the first application were essentially exploiting this property using the laser to ablate tissue, or for example, to photocoagulate tissue, the retina. And initially they were not very successful because people didn't understand very well which wavelength you need and which regime of light matter interaction you need. So this is the first point. I think nowadays people have understood this very well, that you need some specific wavelength, laser wavelength, to interact properly with tissues. And in particular, if you want to do a precise tissue ablation, like in laser surgery, you need to have wavelengths where you have a very high water absorption, because our tissue is 90% made of water. So, for example, this brings you to infrared lasers, like the thulium laser at 2 micron or the erbium laser at 3 micron. This is the first constraint. Another important constraint is the availability of optical fibers, because for many applications, like endoscopic surgery, you don't want to cut the patient, you want to bring the light via an optical fiber. So, for example, for 3 micron, this is not available. There are no good optical fibers at 3 micron that are not toxic for the body. Some of them use chalcogens, which are toxic materials, while for 2 micron, we are lucky, and there are very good optical fibers. So this allows, for example, to use the thulium laser for many surgery applications, such as, for example, when you have prostatic hypertrophy. This is a very common disease in aging males. So you need to reduce the volume of the prostate, and this can be done now in a very non-invasive way using laser surgery with the thulium laser and an optical fiber. So this is a very clean operation. This is one example. Another example is, of course, correction of myopia. So you want to reshape the retina to correct for the excessive curvature that gives rise to myopia. And this nowadays can be done with lasers, in particular with UV laser. In this case, you go to the excimer laser, which allows to really ablate a part of the cornea very precisely to remodel it and to achieve the correct curvature. And actually, maybe just stop me, James, if I'm speaking too much. But another remarkable, I've been working with lasers for more than 30 years, and these very short pulse lasers, so-called femtosecond lasers, initially were very complex laser systems, so that they could only be used in some sophisticated labs. Nowadays, they become very robust, and they even find application in the clinical. For example, for this LASIK operation for the correction of the myopia, what they typically do is they cut what is called the flap in the cornea, and they uncover this flap, and then they do the processing with the excimer laser. But this cut was initially done with a knife by the surgeon. Nowadays, it's done with a femtosecond laser because this guarantees an extremely high precision. So this procedure of corneal of myopia correction, which is done by millions of people every year worldwide, it's now completely done by the laser, which is a remarkable achievement, in my opinion.
Yeah, I mean, you mentioned the start of laser technology being very kind of trial and error in terms of working out how best to apply it. Do you think we've reached this kind of peak understanding of how to apply it now?
I think there is still a lot to understand, and I think lasers are improving by the day. So the progress in the technology is really remarkable, and also I think. These new regimes that were previously unavailable are still needs to be explored. I see many fields in which there is a big room for improvement. For example, just to give you an example, in dentistry, which is, of course, people use still a mechanical drill, but you could do it with a laser, but this is not yet really established. I think establishing, for example, laser dentistry would be a very big beneficial, because maybe it would allow to reduce the pain and increase the accuracy, and it would be a big opportunity, let's say. Yes.
Interesting, yeah. And moving along to Brian now, you've had a little bit of time to think about the question about the biggest challenges you faced. Are there any alternative problems that you've seen, or is it generally more of the same from you, do you think?
No, I mean, the laser is an amazing device. Also worked with lasers for over 30 years. But there are other challenges, I think, in the medical device and biotech industry, right? So one of them is, as Giulio mentioned, is that transparency window in tissue. Now, at a lot of wavelengths, tissue isn't particularly absorptive, but it's very, very highly scattering. And so I think that's the reason why historically we've seen a lot of application of laser radiation to ophthalmology, where the eye is transparent to many different wavelengths of light, right? And where there's a lot of differentiation of wavelength of light absorption, right? So, I mean, one of the first applications was diabetic retinopathy, using the laser's thermal power to sort of seal and photocoagulate bleeding blood vessels in the back of the eye, and stopping the progression of blindness. But some of the other applications, tend to be superficial, so tattoo removal or some laser scalpel work, or else you've got to go to an endoscope, and then you have issues trying to match the laser wavelength to the fibers that you can get, right? I think some of the developments in optical fibers in the last few years, sort of the propagation of so-called holy fibers that guide through air instead of through material, are sort of very exciting in that front. But I also think there's additional challenges in terms of laser-based products. So the cost of goods sold, right? The fundamental cost of many laser systems works for some applications, but can be cost prohibitive for other applications. And the challenge is you not only have to put a device, a laser into a medical device, you got to make that medical device work in a business sense for the models that are going to use it. And a lot of times that's a big challenge. The other thing that's a little different with lasers in terms of medical devices, it's a regulatory burden, right? Not only in terms of getting the medical device cleared or certified, where you have to balance risks with potential benefits, but then also in terms of getting the device onto the market properly certified, understanding the work instructions in the clinic and that sort of thing. So, so the lasers are kind of a little different that way as well, because the light is so bright and so monochromatic and so collimated, so well focusable. You really have to think about some of the safety applications, not only for the patient, but for the clinicians. So you know, I think those are some of the alternate challenges as well when you're bringing lasers to medical devices. Biotech in general, a laboratory device can be operated in more controlled circumstances, but particularly when you think about laser therapeutics or diagnostic instruments, the safety and regulatory issues sort of come in a little bit.
Yeah, I mean, on those safety issues, I think Julio, you mentioned the start of kind of laser eye surgery.
Yeah.
And when it was quite early on to the market, it was seen as quite a dangerous kind of thing to be a part of. But now, of course, we're talking about using lasers for non-invasive procedures which would have previously been invasive. So how do you think perception is changing in terms of laser surgery and for biotech applications? And is that still one of the challenges that has to be got through?
Well, of course, the safety is an issue because, of course, laser can be dangerous, especially for the eye. Let's say this is the main danger. Okay, you can also be burned by a laser, but it's more or less like, you know, touching a hot stove. So it's not terribly bad. I mean, but the eye can be very dangerous. But of course, there are two possibilities. Either you have to use appropriate goggles, safety goggles when you use a laser and be protected. And then you can make what are called class 4 lasers, where essentially the beam is you can never reach the beam as a user. And there is an interlock that blocks the laser if you try to access the beam. So I think all these issues of safety can be solved. Of course, you need to have to reach the proper regime of light-matter interaction, so you don't want to hurt the patient with too much power. But of course, this can be done by properly engineering the product. I think it's also maybe another issue is that, of course, the medical doctor is not an expert in optics. They don't know the physics of laser or the basic. Maybe it's right that they focus on different problems. So they want to see the result. So we need to make the laser different from a laser that we use in our own labs for research. We need to make it very user-friendly and simple. Maybe you have to have, I don't know, some simple buttons that tell you more power, less power, ablate, or maybe coagulate, something that can be easily understood without entering into the physics of the laser, let's say. This is also important, in my opinion.
Yeah, I think it's just to jump in. I think, you know, the doctor needs to spend their cognitive load thinking about the operation, thinking about diagnoses, thinking about the interaction with the patient. As graduate students, you know, we're willing, in my old lab, you'd go in, you know, at 8 or 9 in the morning and nurse the lasers until they were happy at maybe 4 or 5 o'clock in the afternoon, and then take some data. And, you know, that just doesn't cut it for a medical device. You know, time is important in the operating theater, time is important in the clinic, and you really want to make the user interface very straightforward and intuitive so you can minimize the training required and minimize the cognitive load while people are using the medical devices.
Right. I should say at this stage, if you are especially interested in laser safety, not just for biotechnical technology applications, check out our next webcast. Will be specifically on laser safety, so we'll dive more into the subject then. But for now, let's move on to the second question I've got down here. which is the subject of industry academia partnerships. And this will be directed specifically to Giulio first, as your kind of role in academia. You and your team have worked with commercial laser companies before, like Coherent on DNA sequencing, for example. But how did the partnership help you to overcome specific imaging complexities, do you think?
Yes, so I think this cooperation between industry and academia is essential for both sides, because sometimes the academia do a research which is maybe more detached from application, and the industry also needs from the specific expertise of the academia. So maybe this touches now another aspect of laser applications in health, let's say, which is not only surgery, let's say, using the power of the laser, but using the fact that the light is non-invasive. So you can use light to make images or to make diagnostic in a non-invasive and non-destructive way. So if you compare, for example, optical microscopy to other kind of imaging techniques, such as, for example, magnetic resonance imaging, which is very much used in health, the optical technique has a very high, much higher spatial resolution. This is an advantage. On the other hand, if you compare, for example, to electron microscopy can have a much higher spatial resolution, but it is destructive. So the optical approach is non-destructive and with high spatial resolution. So this, I think, is very important. And this is, I think, where the academia can also contribute by developing a lot of sophisticated microscopy techniques. And here, essentially, I see two big classes of applications. So you can use a microscopy for the biologist to study fundamental biological processes. So, for example, if you want to understand at a cellular level the mechanism of a disease, what happens in a cell when a disease happens. So you can really watch live cells and watch interaction of cells with, for example, drugs or other cells. And this is very important for basic understanding. And the other application is the diagnostics. So you can use a laser as a tool for diagnostic. So what is probably the most successful application is at the moment OCT, optical coherence tomography. This is a way to make a diagnosis non-invasive of the retina of the patient. And nowadays OCT is a technique that was developed in the 80s by Jin Fujimoto at MIT, who's an academic, a professor. And now it has become, every ophthalmologist needs to have OCT, because especially for aging people, you can monitor a lot of diseases of the retina, like retinal maculopathy and a lot of, and this can be done in a very non-invasive way. So this is, I think, one very successful example, but there are many others. Actually, later on, maybe I will show also some of our applications that my own spin-off is developing, which is also going in this direction of using light for non-invasive and very accurate imaging.
Thank you. And Brian, did you have anything that you wanted to add about that kind of industry academia partnerships? Have you been involved in any in the past?
We've been involved in some sort of consortium partnership. So I should explain, Starfish Medical, for whom I work, we're a design consultancy organization. So people would come to us with an idea for a medical device or a problem that they want to solve, and ask our team of engineers, mechanical, regulatory, safety, human factors, ID, optics, to help build that device. And so for us, a lot of times, what we bring to the mix in sort of partnering with academic partners, and we've done a number over the years, is sort of understanding that business aspect, as well as having the product expertise. Because as I sort of alluded to earlier, getting a laser system to work in the lab is not the same thing as packaging it up, making 100 of them that are identical to each other, shipping them across the continent or across the ocean, and then having them work in a robust way. So there's that synergy between the the ideas and the fundamental knowledge that one finds in academia and the creativity, right, of thinking of new applications and wanting to ask, what else can I do? What else can I see with this? And then marrying that with the discipline and the constraints of product engineering. And so that's really, and it's not like the academics have no idea on how to build something in a cost-effective way. And it's not like our engineers have no idea how to do something creatively. We pride ourselves on that. But there's that sort of natural give and take between the two sides of the partnership.
Yeah, yeah. Do you think it kind of it's important to see both those sides of the coin from the start of a project or of research, or can it kind of stifle that kind of creativity?
Um, Sorry, I'll just jump in. I think, I mean, you need the creativity, right? And so you need those ideas to come from somewhere. But if you're waiting too long to bring in the other aspects of product development, you run a risk of not being successful. Because, when I transitioned from academia to industry, one of the things I had to learn was, your technology is not a product. Your product is technology that works in a robust, repeatable, manufacturable, cost-effective way, and that people will buy. And so there's quite a lot just beyond the technology that really determines whether you'll be successful in getting your technology into helping people and improving their lives, or whether it just won't work out. It's for the most part a capitalist world.
Yeah, I think as Brian said very well, we need both these aspects. Maybe you need the intellectual creativity, also maybe taking the risk to develop something new that may or not work. And this is typically done in academia because you do frontier research. But then you need to translate this into a product. And I think it's a slightly different set of skills. But maybe having a common cooperation and a mutual respect between these two aspects is very important, because actually, I must say, I've been doing for many years basic research without, let's say, having a product in mind, which is very nice. But now, recently, also my younger students have founded some spin-offs that are coming from my lab that are actually quite successful. And then I see they are, of course, 100% focused on getting a product, because if they don't get a product, they won't survive, basically. While, I mean, I can always apply for some grant, but they need really, and I think it's very nice. But I contribute to this maybe with some new ideas, and that's very, very satisfying, I think, because then I see also some of my ideas coming to becoming real products and being used by people. This is really nice for an academic to have this aspect.
Yeah. Moving on now to another question about the applications of laser technology in different sectors. There are many varying applications. And medical science is just one. But Brian, specifically, you shifted from semiconductor manufacturing into the world of biomedical production. What are the standout differences for you, do you think, between those two sectors when comparing laser applications and what they involve, do you think?
Well, particularly when I worked for Cymer, which is now part of ASML, the market was very well defined. In semiconductor lithography, there is no way to correct for chromatic aberration, so it all becomes about extremely narrow wavelength, narrow bandwidth laser sources, and you want to write small features, so it's got to be in the ultraviolet. And Cymer has been building lasers for lithography since 1986, so the market was very well defined. Um, and the laser was essentially a very high powered light bulb with very good spectral qualities. Moving to the biomedical industry, and particularly in a consulting firm, one of the big distinctions is really the variety. Sometimes for us, the laser is a light bulb, sometimes it is a probe, sometimes it is a scalpel. And it, the range of wavelengths can vary from the ultraviolet for sterilization, let's say, all the way to the mid infrared. So there's quite a lot of diversity in how you use the laser and how you think of it in the medical device sector. The other big one is just the overriding integration of risk analysis into the design effort, where you're always trying to balance the benefits with the risks and trying to ascertain those things. And you're often folding that into the design process and trying to understand what's the largest amount of power that I can get away with and still maintain classification, or do I have to go to a different class? How does that control the system? If you're making a medical device, you have to make sure that it's safe and effective. So you're thinking in much more detail about failure scenarios and how to mitigate them. So that's, all of the lasers that Cymer made, of course, went through qualification testing, right, and safety testing, but the product was much better defined. And here you're sort of really integrating those aspects into the design constraints in a different way. And the final thing that I think is, a lot different between semiconductor lithography and medical devices is in semiconductor lithography, you control all of the optics, all of the way onto the silicon, and including the silicon, and you know a lot about it. You might not know the detailed, illumination patterns or those sorts of proprietary things, but you understand the optics all the way through. In any medical device, after your beautiful optics shape your laser beam and make it exactly right, bring it to the right location, then you have to go through your final lens, which is the human body. And so you have to think in ophthalmology of the physiological variation in your patient population. And that eye, unless you're applanating, is a strong focusing lens. If you're going into the skin, you have to think about the scattering. And then you have to think about the difference in scattering and absorption for different people, for people from different genetic backgrounds and that sort of thing. So it really sort of broadens up the design space, and it brings a lot more challenges, but it brings a lot more stimulation, if you will.
Okay. And Julian, from your side, has your work always been focused on biotech, or have you done other research in other areas that you've been able to apply here?
No, well, let's say my research is mainly focused on nonlinear optics, so exploiting nonlinear optical process to generate light pulses of different colors and then their application. So the biotech is just one part. For sure, what I can tell is that if you go to the medical application, especially let's say the real surgery, the going with patients, or even the diagnostic, it is a very long process to develop a product, because I guess if you use a laser for materials processing, as long as the outcome is satisfactory, you can sell the product. But if you want to have a laser for surgery, then you need to have to go through a lot of clinical trials and get the product approved. So this may take years and years, for a good reason, because in the end you have to go to a patient, so you have to be sure of the result. So this is, I think you need also probably to incorporate all these elements in your design from the beginning. So I think if a laser for material processing fails, okay, you get some bad products, but if a laser fails during surgery, you may get a very severe damage. So also the design must have this in mind, and then you must be ready to invest a lot of time until you have a product. So the development phase is much longer. This also may be a problem potentially for some investors, because this will require to wait a bit more until generating revenues on a specific medical product.
Right, of course.
Yeah.
On the subject of working with commercial clients, then Brian, when medtech partners come to Starfish, are there any common pain points that they tend to have, that they tend to bring to you, and how do you kind of tailor the use of lasers to meet specific varying kind of specific needs?
Yeah, I think a lot of times, many of our initial discussions are helping the client sort of understand the possibilities of lasers, because lasers offer a lot of advantages, but they also offer a number of challenges. Julio mentioned that, you got to be able to get the laser to where you want to treat it. And so you have to have the optical fibers, and that can constrain very much the light source that you're using. And that's just one example of many. But oftentimes, whether people want to integrate a laser in their device or they have some other technology, a lot of our initial discussions are about, first of all, helping them to define what their product is, because it's not enough to have a great scalpel. You got to have a great scalpel that cuts, that reduces post-surgery complications, or that enables you to do things faster. At least in the North American market, if you can't provide the service, within existing sort of bill codes for Medicare, that's what a lot of things drive, then the product won't be taken up by the medical profession, because it just doesn't make cost sense for them. It's kind of sad to think about things that way, but it's important to understand that going into the whole effort, especially if you're in a place where people have fixed remuneration for medical procedures. The other thing that we often try to get people to understand, and Julio sort of mentioned this in passing, right, is you want to sort of sculpt your development process to make sure that you're able to raise the next round of funding, because oftentimes it takes multiple rounds of funding to make a medical device. And sometimes we help people clarify this great product that they have in their mind, which we are on a journey to make. But in the meantime, maybe there are more important things to do, like have this aspect of the device understood, have a rendering of the device to help investors sort of feel it here as well as, analyze it here. And so a lot of times we're helping people think about the development process and how to spend your money so it has the largest long-term impact and you can sort of preserve your ownership stake. And those sorts of questions and conversations are often coming when we have people who are coming from academia with an idea, or people coming who are medical doctors who have an idea for some technology. We sort of explore that idea of a product a little bit more, and more so, we're not neither we nor our partners are really product companies yet, right? We're developing products. And so we have to think about that cadence of fundraising and minimum viable product for the next big raise and that sort of thing. That's sort of universal across the medical device application or technology.
Yeah, yeah. And in terms of funding then, Julio, how do you go about sort of pitching projects and studies to get that funding? How are you inspired by kind of what applications you're going to look into?
Well, let's say now I can talk for my specific company. Actually, we have a big dream, which is, say, to revolutionize histopathology, so that the diagnosis of biopsies. But this will be a long journey, as Brian was saying. So in the meanwhile, we think that our technology, which is a coherent Raman microscopy, can have many low-hanging fruits that we can apply to. So first of all, to scientific research. So many people could you benefit from laser technology, not only for, say, clinical application, but also for scientific. And in the scientific application, it's much easier because you don't need to have a product certified to go on a patient. It needs to be, of course, certified for safety, but this is much easier. And this can be first. And then there are other applications, for example, that can be spin out from the medical, where if you want to detect the composition of, like, instead of looking at the composition of a tissue, we would look at the composition of other substances, for example, of a drug tablet. So doing quality control in the pharma industry, this could be another big application that we are looking into. So this, for example, of course, doesn't need to go through all this approval path length, which may be quite complicated and longer. So I think, but of course, we also keep in mind our big dream, which is to go finally to the patient.
Yeah, yeah, of course. And sticking with you, Julio, then, from your point of view, as I think as co-founder of Cambridge Raman Imaging, you're aiming to boost the efficiency of histopathology and precision imaging applications using a microscope. How useful will lasers be there?
Okay, so they will be essential. So if you allow, I prepared a few slides to present what we are doing. If you don't mind, I will share them briefly.
No, please, yeah.
Yes, let me see. Okay. So actually, the laser is essential for our application. In fact, our laser, we are developing, actually this is in the framework also of a European project, of an EIC transition project of the European Innovation Council. And so this project is called CHARM. And this is actually led by Matteo Negro, who is the CEO of our company. And the company was founded by me together with Andrea Ferrari from the University of Cambridge, and it's called Cambridge Raman Imaging. So essentially, we start from a very important process that is done on billions of samples every year, which is essentially histopathology. So you take a biopsy of a tissue, and what is still done nowadays, after more than 100 years, you go to this tissue, you stain it with two dyes, actually hematoxylin and eosin, that's called H&E staining. And then the tissue becomes colored. And until maybe 20 years ago, the standard was that the medical doctor, the histopathologist, would take the slide, put it under the microscope and look at the cells, and then looking at what they see, make the diagnosis. This is healthy, this is a tumor, what kind of tumor this is. So of course, this is a very time-consuming and also kind of subjective procedure. So there was a first revolution a few years ago with the introduction of digital pathology. So in digital pathology, what you do, you have the same slide, the same tissue sample, still colored with dyes, but now instead of putting it under the microscope, you put it into a machine, which is the digital scanner, which takes a high-resolution image of the tissue. So you look now on a screen of a computer, you can zoom in and look what you would see under the microscope. And this, of course, the availability of a digital images has many advantages. And of course, the most important is that now you can rely on AI modules to really analyze the image and support the diagnosis. But let's say all this doesn't tell you much about the chemical composition of the tissue. It is essentially telling you about the morphology, about the shape of the cells. So what you can do now, where the laser can help, is by getting information on the chemical composition. And this you can do with so-called with spontaneous Raman scattering. So essentially in Raman scattering, you look at the fingerprint, the chemical signature, which is due to the vibrations of the atoms within, let's say, within the molecule that make the tissue. And these are characteristic frequencies that basically identify a specific component, like carbohydrates, lipids, proteins, DNA. All these have a characteristic Raman spectrum. And so you can use now spontaneous Raman to identify tumors. This is a collection of results. It can be very specific, with very high specificity and very high sensitivity, meaning that you only identify the tumors and there's no false positives. However, this approach is very slow, so you cannot really apply it. So that motivates to go from spontaneous Raman, where you have a single laser and the molecules vibrate each independently from each other. So these are spontaneous random thermal vibration, and you interact with these molecules and you get the signal. So what you can do is now you can do coherent Raman, where instead of having one laser, you have two lasers, and these two lasers have different colors, and these colors now drive all the molecules to vibrate simultaneously. So they generate a vibrational coherence. And this has a dramatic effect because it increases the signal by 5-6 orders of magnitude. So now you can really make these images of these chemical images very fast in a time that is reasonable for an application. So this is basically our motivation to develop such a laser in our company for doing this coherent Raman microscopy. And actually, let me skip some technical details. Nowadays, the lasers that are used, even in my lab, are these big, bulky machines that are good for scientists, but you cannot give them in the hand of a doctor because they are simply too complicated. So our idea was to drastically simplify the architecture, making a fiber laser. So now we have a very compact fiber laser that, and we have a way that we have two lasers that we are synchronizing them. So these two lasers, these two fibers generate light at two different colors. and we can now use them for doing this microscopy, this vibration coherent gamma microscopy. And actually, we have engineered this into a very compact and turnkey product. And so this is essentially a box when there is nothing that you can adjust. Essentially, it is turnkey, and it's like a big microwave oven, let's say. It's something very compact. And now with this box and some additional electronics that we have developed, we can really, we have this microscope that is now really taught for going into the clinic and being used on biopsies. And just to give you some examples, this is like, we can do what is called AI staining. So we take a tissue which has not been stained by any color, and we can generate, using our technique plus AI, the same image that you would get with the staining. So, and we can do this in a few minutes, while the staining process takes multiple hours. And we can do this on this is a real clinical biopsy taken from a head and neck cancer tissue. And this is like a centimeter square. And we can really make, with a very high speed and very high, this chemical image that really tells us about the chemical composition. And let's say our long-term dream will be a chemometric tumor diagnosis. So we would like to acquire for a patient, for a tissue slide, without doing any staining, any chemical processing, just the tissue as it is. We would like to acquire an image where we get the Raman spectrum for every pixel, for every point of the image. Then we get a so-called hypercube, because we have many. We have a spectrum for every pixel, and then we can use AI. We can use automatic analysis, and we can train these on many. And the goal would be that in the end we identify the tumor. This is the first goal. But then also, not only we say this is healthy or sick, but we also give a very precise identification of the type of tumor. So we do molecular subtyping, and also we do grading. So what is the level, the grade of the tumor, low grade, high grade. And this is essential information for the surgeon to understand what to do, what is the best therapy. So our dream would be to use this machine for what is called personalized medicine, so that you can really make a very precise, the best treatment possible for the patient according to the identification of the disease. So this is, of course, our long-term dream, but and this requires the laser, because we had to develop a very special laser to do this. And now I'm very happy that it's working in the lab, that we can generate this data. And so now, of course, it's a longer road until we can really apply it to patients. But meanwhile, as I said, we have these other applications which appear more feasible.
That's really cool.
Right. Thank you for that, Julia. That's good. That's good. That gives a great idea of the technology and the extent of the usefulness there. The next question I was going to go on to is about kind of the advice that you'd give people within the industry. So that it's a great time to bring in the first question we've had from our audience here, which is someone's asking, as a postdoc researcher, am I able to start an independent company with ideas that I developed during my research, even if my idea is different but related to the research topic, or do I have to involve the institution in that? I guess that's Julia, you'd be best to.
Okay, so I think, of course, as a postdoc, if you have developed something, for example, I saw some of my PhD students or even master students who developed some technology during their thesis, and then they decided to found a company. So there is no need to involve your institution, unless there may be some patents that you may need to acquire. But apart from this, actually, I think for example, my institution, particularly Milano, helps the students. They have a program for people who want, it's called Switch to Product. So the idea is that they train maybe students who have very good ideas but no experience in founding a company, and they couple a mentor to them. So for example, in one of our spin-offs, we had a very good mentor who founded multiple companies. He got a very small fraction of the shares, And for this, he provides a lot of advice, also a stupid thing like what is the product that you, what is the price of your product? How do you determine? I mean, this is something that maybe a scientist is not obvious, how you should calculate a good price. And so I think our university is very keen on promoting these initiatives. And so in this sense, it is something that is actually, you rather than getting obstacles, you should get benefit from the university.
I think if I can jump in,
Yep.
it may involve a bit of a delicate dance as well. And certainly universities are very interesting in developing industrial output, not only from the success of their research community, but also, a lot of granting agencies are very interested in, oh, well, how much money is it going to make in the long term? Interested to tie these linkages, right, to the research that they do that are answering fundamental questions to products that are on the market. I just say some North American universities can be very aggressive about maintaining some intellectual property ownership for the licensing fees, right? They use that, particularly in the States, as a source of additional revenue when state funding isn't that high. So I think it might make sense to look a little bit more into the contracts you've signed as a postdoc and what it says about intellectual property, and then maybe cautiously reach out to the industrial liaison or the incubator for that particular university. Because I think, yeah, as Giulio says, you've never made a company before, but the university has probably helped a lot of people from the university community make companies, and you can leverage that. You also just want to be very careful, I think, about making sure you understand your intellectual property, whether the university has any share in ownership, and how aggressive they are, because that can vary a lot, I think, from institution to institution.
Right. Interesting question and interesting answers there. So thank you for both of those. Cautious optimism, I think, is the key there. So moving on to the other bit of advice that you might have. Let's say I'm an engineer or a product manager this time, who's new to the biotech sector or the medicine sector, and I'm thinking about using laser-based application. What are some of the top tips or the kind of best practices that you can give me as advice? Do you think maybe, Brian, if I can ask you to kick off us on that one?
Sure. I think maybe I've covered some of these sort of in passing before, but the first thing I think I would advise people is understand where your product fits in the market and what the commercial success is likely to be. I mentioned things about the North American market and how that's dominated by how much Medicare in the States will reimburse a doctor for a particular procedure. And whatever product you're using has to fit into that cost structure in order for doctors to take it up. Now, they might take a lower profit per procedure if they could do more procedures in a day, but you really have to go in asking, what's the product? How does it fit in the marketplace? The other super important thing for medical devices is to understand the regulatory pathway and the markets that you want to go into. And the requirements for certification and the different classifications of device are different in Europe and in Japan and in Canada and in the United States. And particularly in Europe, certifying bodies will look favorably upon FDA certification. And I think the converse is also true. And in the end, all of these certifying bodies or approving bodies are looking for that sort of risk analysis and understanding, what's the benefits, what are the risks, how are you managing the risks? And getting on that early can actually make the difference between sort of a product that was dead on arrival versus one that can be successful. And the only other thing I'd say is work with your stakeholders to understand what the next important milestone is and what's the simplest way to sort of de-risk to that milestone. And sometimes it can be a little bit of a parallel path to the eventual product, but you really want to spend your time and your effort wisely.
Right. Thank you. And Giulio, do you have any kind of best practices advice yourself?
Maybe, I mean, one important point coming from the perspective of somebody in the academia who teaches optics and lasers
Yeah.
Maybe it's important to learn the basics of laser physics and optics, because I find that this is something that is not, there are not many people who are really expert to understand very well these things. So if you become, I think, as an advice to a student, if you become a real expert in lasers and optics, you will never be out of job, because they will always look for you. And then the other aspect is you need to understand, if you want to go into the medical application, also a bit of the bio part. So you need to understand, to know a little bit of biology. Of course, you don't become a medical doctor or a biologist, but you need to be able to talk to them. And this is very interdisciplinary, because and you need to also look at the basics, let's say, of light matter interaction, understand it, how light matter interaction works, and so in order to properly apply your laser. So I think having this kind of cultural background is very important, that will allow you to talk also to your end users.
If I can just jump in, there's one kind of little technical tidbit that's worth remembering, which is, you know, lasers are very bright, and so that's one sort of bit of understanding that people struggle to really understand, because a laser light is radically different than any other light source, you know, in the solar system. But the other thing is that the coherence, the monochromaticity, and the regular phase relationship between different photons in a laser beam, it can be very powerful. For example, you know, with what Giulio said, his technique gets a factor, you know, that grows quadratically with the number of molecules, because he's using coherent excitation. But it can also be a royal pain in the neck, because if you're trying to use a laser to treat tissue, and you're piping it through a multi-mode fiber, or if there's any scattering on the way, you're going to get laser speckle, which can induce up to 100% modulation across the beam footprint, right, in the irradiance and intensity. And so then you have to worry that your dead spots are too dead and your bright spots are way too bright. It's not enough to just think about average irradiance. And that's kind of a little subtlety that can sometimes bite people.
Right, right. I'm going to try and just quickly slip one more question in before the end. How do you see, maybe I'll go to Giulio first with this one. How do you see laser applications evolving across the sectors over the next few years? What are some of the really groundbreaking technologies, innovations that will give us that kind of wow moment, you think?
So I think there are many things. So I think the future looks bright. I mean, as the lasers are bright, also the future of lasers is bright in the application to health. So there will, I think on imaging, on the side of imaging, there will be more and more sophisticated imaging techniques. And now, for example, I've shown you ex vivo technique, where you, for example, you take a biopsy from a patient. But of course, you can think of in vivo imaging, which you don't need to take the biopsy, which of course will be even nicer. And so this can be used to improve the diagnosis, but also maybe perform a diagnosis during treatment. For example, this is what's called intraoperatory diagnosis. For example, you are operating a brain tumor, you have the patient in the operating theater, and you want to take a sample of the tumor and immediately in a few minutes to get, let's say, a diagnosis. And this can be done with these new technologies that are being developed. This, of course, would be a huge improvement. And of course, now I was mentioning femtosecond lasers. I like them a lot because this is my main field of research. So when I started working on them 30 years ago, there were only a few labs in the world who could make these lasers. Now they have become very reliable. So thanks to the very short duration, they can really allow new kind of therapies. And especially for surgical application, like in the eye surgery or other applications, they can be very, very interesting. And finally, I think there is another topic which is very important, which is nanomedicine. So using some nanoparticles or some like different either organic or organic nanoparticles to carry, for example, a drug and to let it arrive at the cell, either to maybe destroy a tumor cell or maybe to heal and to perform some other kind of treatment. And now, for example, you can use the light to activate the drug release. So there is this nanocarrier that brings the drugs into the body to where it needs to go, and then you bring also some light that opens this cage and releases the drug. This is, of course, some more science fiction application, but very, very fascinating for the future, I would say.
I think there's also just the thermal applications in nanoparticles, right? That can be very highly absorbing, and so even in a more brute force manner, I think you put your finger on some of the paths of future treatments, Julia.
Yeah, I think all great kind of innovations really can start off in science fiction. There's no reason not to bring them into reality a lot of the time. But that sounds really great and exciting for the future. I think with that, that's just about all we've got time for. So I'd just like to say a big thank you again to our guest speakers, Julio and Brian, for taking the time to be with us today. As I mentioned earlier, the web panel will be available on demand in the webcast section of the Electro Optics website, along with previous recordings from the past 12 months. So please do go and check those out. And as I mentioned earlier on as well, the next discussion we've got lined up in the Photonics 100 web panel series will be live on Wednesday, the 30th of April at 3 p.m. And I'll be back here discussing the best practices for safe and compliant laser operation with our next panel of guest speakers, which will include Scott Wolstein, who's president of the Photonics Group, David Lawton, a market development manager at Kentech, and Federico Bonaldo, who's a physicist in the R&I department at Castellini. So I'm very much looking forward to that. Until then, however, I hope you enjoyed today's discussion and have a good rest of the day. Goodbye.
Thank you all for your attention and your questions.
Bye to everybody. Thank you.
Thanks a lot, gents.